morning, everyone, and welcome to the MMID Coding Workshop series. So the instructor for today is Taylor Davedow, and she's in her second year of PhD studies at the University of Manitoba in the Department of Medical Microbiology and Infectious Diseases, and she's being supervised by Dr. Celine Nadon. So today, Taylor is going to provide us with an introduction to ggTree, where she's going to focus on how to import the tree data, as well as build some basic skills in visualization and annotation of trees. All right, Taylor, the floor is yours.
Thanks, Jill. Hi, everyone. Welcome to today's presentation in the workshop series. Today, we'll be focusing on data visualization using ggTree and just give a very basic overview of some of the steps that I usually do when I'm just working with a tree. So just a reminder that all the workshops are being recorded and will be posted to the MMID Coding Workshop YouTube channel. There is a Q&A session at the end of today's workshop, but it will not be recorded. The objectives for today are to learn to install and load some packages that we'll be using into RStudio, load a NUIC file and complement files, learn how to create a basic tree, changing the layout of our tree, adding and customizing the tip labels, learning how to merge our tree with a heat map, and finally exporting the tree. The data used in today's workshop was modified from a genomics study about Vibrio cholerae that was published in 2020 by Arteaga. We will only be working with results on a subset of 26 samples, whereas the original paper had 70 samples they were working with. The workshop materials were uploaded to the MMID Coding Workshop GitHub repository and can be downloaded if you want to follow along or if you want to try it on your own time. The files we'll be using include a tree file in NUIC format. This is one of the standard formats for representing trees in computer readable form, although there are other formats that ggTree can read in. The NUIC file was created by me after downloading the genomes listed in the manuscript from the previous slide and running the FASTQ files through the Snibble pipeline in Galaxy. The metadata file is in Excel format, and I compiled it based on information that was given in the manuscript, as well as the supplementary materials. And then the nucleotide blast output was just simulated by a random number generator to produce a column of percent identity, just for the purpose of this workshop. So it does not accurately reflect the results of this study. So just keep that in mind. And also there are 7 genes that will be included instead of the original 37. The original figure in the paper looks like this. It includes a phylogenetic tree based on whole genome SNPs. They also used an R tool called RIRBAPS for grouping the strains into these 5 sequence clusters, as well as 12 lineages. We will not be using this tool, but if you're interested in, expanding and trying to make a tree more similar like this, definitely look into it. It's described in the paper. It also includes this middle section with some of the metadata for each isolate, and then finally the heat map. So the heat map, they defined the presence and variation of each gene using nucleotide identity levels ranging from 80 to 100% using this color intensity scale, and then everything lower than that was shown as gray. What we'll be creating contains some of the same elements, just a little bit more basic. So it's got the tree element, the tip labels with a little bit of the metadata information, and a heat map. ggTree is a package for R programming language, and it was released under the Bioconductor project. It was created by Dr. Yu for visualization and annotation of phylogenetic trees and other tree-like structures in conjunction with their annotation data. I highly recommend checking out this book that was written by the author, and it contains 13 chapters of just really good information and tutorials. Although there are some freely available online tools, ggTree is really robust and programmable, which is what we're looking for in these workshops. And it allows for a high level of customizability for different aspects of your tree data. So let's jump into the first objective, which includes installing and loading the packages into RStudio. We'll be using the following packages. So first off, we have readxl, and this will be useful for reading in the XL files. This is the metadata and blast files. The BioC Manager package will be useful for installing ggTree afterwards. The tree.io package is mentioned a lot in the ggTree book, and we'll be using it for reading in the Newick tree. But phytools I've mostly used previously, and it has the same function. And it also will be used for midpoint root. So I added them both there as options. The tidyverse package will use for tidying, a little bit of data tidying, and ggTree, of course, for tree visualization and annotation. And then ggplot2 will use for some additional plotting support. There are a few different methods for installing packages that are listed in the R script as part of the workshop materials. I used install.packages with the package name in quotes for the packages listed in the previous slide. For ggTree, we will need to use the BioC Manager to install as listed there. To load in the packages, we use library with the package name in quotes. And just a note that package installation usually only has to be done the one time, and then each time you want to use a new library, you'll have to load it in. Next, we're going to load in our files. So as I mentioned, we have 3 files to load in today, and we will view and possibly tidy some of them later on. The first one is the tree file, and we'll use read.newick with the sample location in quotes. And then I assigned it to an object called tree. The metadata and blast files can be read in using the read_xlsx with the file location. I think these slides are a little bit different from what was uploaded. Originally, I used the here package. If you want to still use that, just make sure that you download that, but I can get Grace to upload these new slides. Next, we're going to learn how to create a basic tree. So at its most basic form, the function is ggTree with the tree in brackets, and we called that tree. So just ggTree, tree, and this is our basic output. If you want to add other arguments, there's a whole list of different arguments to expand on what we're talking about today. You can look at the ggTree help page as well. And then we can change the layout. So the standard layout is rectangular. And to specify layout, you just do layout equals and then the shape in quotes. I think there's about 12 different shapes. Since rectangular is the default, you don't actually need to specify it. It'll show up automatically. But you can also have circular, slanted, it all depends on what you're looking for. You can also use the branch.length equals none. And this will set the length as none, basically to make it look like a cladogram. Opposed to if you don't include this, the default will still be the normal scaled branches as shown in the rectangular. The midpoint root roots the tree at the midpoint of the longest point between the 2 tips. For this demonstration moving forward, I'm going to use midpoint root because that's what the authors had used. But also you should keep in mind that the type of root and position you choose may drastically change the topology of your tree, and you should make sure you're choosing an appropriate method. This is something I'm still learning about and need to read more about as part of my own research, but just something to keep in mind. By adding these internal node numbers, we can see how we can manipulate the tree based on these node numbers. So you don't need to keep them in your tree, but they're good to add so that you can reference them. So to add them, we use gm_text2, followed by this code. And then you can see that they're listed here. So moving forward, I'm going to show some examples based off of referring to node 41. So just keep an eye on that. If we pipe our tree into the collapse function, as shown in the second square, and we specify node 41, you can see that that node collapsed. It's no longer there. If we use scale clade and specify node 41, setting the scale to anything less than 1 will shrink the scale. So that clade is shrunk. And if we had set it to anything over 1, then it would expand the clade. There's also the flip function. So we can flip these two clades, and now we can see that clade at node 41 is now moved. So if you want to change the positioning of your tree. To view or zoom into a particular clade, that can be very useful. I actually didn't notice the difference between using the two different operators. So if you use pipe and specify a certain node, it'll just zoom into that particular clade. But if you use the plus operator, it'll show the original tree next to the zoomed in tree. So you've got different options there. Next, we're going to add and customize some tip labels. So if we want to check what our tip labels are going to be ahead of time, so we know what to expect, we can use the head function and specify our tree, which we called tree, and say tip label. And we can see here that it gives us an overview in the console of what it's going to look like. And right now our tip labels are the SRA numbers from NCBI. And these were the names of the FASTQ files when I had run them through Snibble. And so that's why they are the tip labels. And so if we set up our tree and using the midpoint root, and we add the tip labels, this is what is going to appear as the tip labels. And I also added a couple other features to help with visualization. I added gm_tree scale, and I set the X and Y coordinates both as 0. So the scale will show at the bottom corner. I also set the width of the scale as 0.01. And then I added two other things here to help visualize the plot. So I added this chord partisan, and this is a coordinate system. Normally there are limits that are set on the plot. And so by turning this clip off, it allows us to draw outside the plot, and it's not going to cut off our tip labels because they're pretty long. Also, I set a plot margin around the tree, and it just gives you extra space around the tree as well. So that's a lot of the defaults that I'll be adding on most of the trees moving forward. So this is the same code as the previous slide. And as we can see, we've got our tip labels. But these labels have very little meaning to me, so we can change them to something more meaningful, such as strain ID, or for your own research, maybe your sample number, something that will be more meaningful to you. But first, in order to change the tip labels, what we're going to do, there we go. What we are going to do is we're going to need to link the metadata file to the tree. And so we need to make sure that there is a way to link the two files. And so we need a variable that will link between them. The tip label names shown here will need to link to the metadata file, and there is a variable called file name. And so this is what is going to link the tree with the metadata file. And we're going to want to instead call it strain ID. So this is going to be the linker. So besides just viewing these two files, we can also create a vector that will check if there are any file names in the metadata file that don't match any of the tip labels. So hopefully that makes sense. And this operator here, it's like %sign in %sign. It will take a vector as the input to the left, which is the tip label, and it will evaluate each element to see if there is a match in the vector that follows on the right, which is the metadata, and then we specified file name. So in this case, if we run that in the console, we got this output, which says there's 0 characters. And so for us, that means that all 26 observations in the tree match all 26 of our observations in the metadata file. So that's good. There has been instances where my metadata file hasn't matched up to the tree. And then you'll see in the console an output of the list of the file names, for example, that aren't matching up. And so if they don't properly link, you're not going to, I don't think the tree will properly produce. So this is even sensitive to spaces and I think case sensitive. So now we're going to customize our labels by linking the metadata file. This operator here is used to attach the annotation data to the tree. And it's also discussed in the ggTree book if you want to read more about it. So we have the tree and its midpoint root as before. And then we add this operator, and then the name of our metadata file object is called metadata. So we added it here. And then the tree scale at the chord partisan and the theme is the same as before. And then we added the geome tip lab as well, but this time we specified the label as strain ID. And so what happens is that now we have the strain IDs appearing instead of the SRA numbers. So for your research, that might be a little bit more meaningful. We can also add a few more features to make the tip labels easier to read. For example, the only thing I changed here was the color. So maybe blue isn't more easily readable, but I changed it to blue arbitrarily. I increased the size to 4 because I think the default is 3. I also added a offset. So this offset will make sure that the labels aren't clinged right to the tips of the tree. It gives it a little bit of space. And then I set align to true. So this makes the labels all aligned. And then it also creates a dotted leader line, which I think looks nice. And so it looks something like this. So you can see that they're all aligned, so it's a lot easier to read. This is the daughter leader line I was talking about. The scale is there in the bottom left corner. So it's starting to look pretty good. And I also assigned it to a new object called ggSimple because I'm satisfied with this as our basic tree. You can keep adding layers and layers of geome tip labs as they had in the journal, the way that they had their metadata kind of all in columns. So you can keep doing that for as many layers as you want, but you'll just have to keep offsetting them so they're not overlapping. So we are going to add another tip label. And this time we're going to add a layer with the Sierra group information. I just set the color as black, and this time I added an offset of 0.05, and that's so they don't overlap with the strain ID. Everything else is the same size, and I aligned it as well. But this time I added a new argument called line type equals NA, and this will remove that dotted leader line And you'll see why in the next slide. So if you don't offset it, this is what you'll get. If you want to do this right away, and then you can kind of gauge how much offset you actually need. Sometimes you need to play around with it. It could be a little tedious, but it works. And then once we offset it and align it, it looks like this, but those dotted leader lines are kind of cutting right through the strain ID labels, and it just is not easy to read. So that's why we set line type equals NA. And it takes away the leader line between those two. So now it looks a lot better and it's easier to read. I really suggest kind of playing around when you're learning these new functions, and then you can see what they do, because it's really easy to arbitrarily add things that don't really make a difference, or add things that you don't really know what they do, and it can make your code really, really long. So next, we're going to look at how to merge the tree with the heat map information. We will do this by using gheatmap. It's a function within ggTree that allows us to join a heat map matrix to a phylogenetic tree. At its most basic form, it looks like this. The P that they have here, that will be our GG simple, the tree that we already saved. And then this data is the matrix or data frame that includes our heat map information. So 3 things we need to do to prepare so the BLAST file will now merge with the tree. First, we're going to just tidy it up a little bit. We're going to set the row names to the file name so that they match up. And then we're going to select out our columns of interest. So we want to tidy up the BLAST raw file that we had saved and change it to a data frame. And then we're going to save it as a new object called blast_df for data frame, just so that we're not, we're going to keep the raw original just in case we need to go back to it for any reason. And I also filtered the %identical variable for greater than or equal to the 80% as the authors had used. I also used pivot wider to widen the data. So originally it looks like this when you view it. And this way I chose names from the gene names. So now they're created as columns. And then the values from %identical are here. And the anything less than 80 will appear as NA. And then I piped it through as a data frame. If you don't change it to a data frame, there is an error that happens in the next step. I also use the relocate function just because for my own visualization purposes, I want this particular gene to appear first. Next, we're going to set the row names so that they match up with our tree. If we want to check what the row names are before, we can do row names and our BLAST data frame, and we can see that they're 1, 2, 3, 4, 5. And you can also view it in the console. And to set them, we do row names and we specify that we want it to be the file name. And when we check it after, we can now see, okay, yes, it's those SRA numbers that links to the tree. But now we have the row names and the file name variable, but we don't want this. It's going to interfere with our heat map. So what we do next is we just select out the file name. So we just are left with the genes. So if you view it before, this is what we had in the last slide. And now we're going to select everything except the file name. So we're just left with the genes. So it's in the perfect format that we want. And we can try making our first heat map. So I specified GG simple, which was our original simple tree, and this BLAST data frame, and something pops up, which is good. But you can see that everything is overlapping. So based off of what we saw before, it looks like we need to add an offset. So when you add an offset of just 0.1, it kind of pushes the heat map off of the labels. And if you have more layers of metadata for tip labels, you'll have to just offset it a little bit more. So that might take some playing around with. And then there's also a width function. This will change the width of the heat map in respect to the tree. I think the default is 1, and I just made it 1.5. Next, we can adjust the column labels. So and we'll do this by using call names position. So that refers to these labels up here. They appeared automatically but they were originally at the bottom, and I just think they look better at the top. So I set call names position to top, and that's the only two options, is top or bottom. I increased the font size of those column, the gene names a little bit. And you can also do other things to call names. I think you can turn them at an angle, you can offset them, you can add different levels. There's tons of different options. The final thing that we're going to do to spruce up our tree heat map a little bit is to adjust the colors. So I will show you two different options. There may be more. The first is going to change the heat map colors directly in heat map using the arguments low and high. So by adding low, this specifies the color at the lowest value, so closer to the 80%. And high will specify the higher value, so 100% as dark blue. There is a color argument here, but that just refers to the cell borders. And all the NA values will remain white, and to my knowledge in gheatmap, you can't change that. So it'll look something like this, a little bit closer to what we were looking at the beginning. Of note, I also had to change the legend title right in gheatmap. So I changed it just to %identity. And I think everything else is the same. There is another way to change the heat map, and that is through ggplot using a scale fill type argument. I used scale fill gradient. Although you can also try scale fill viridis if you want to use more accessible colors for visual impairment. ggplot doesn't like us to use these multiple fill scales, so there will be a warning message that's saying it's overriding the previous scale, but it'll just use the most recent scale, and it'll still work. And again, I used scale fill gradient, and I specified the low and high colors. I also had to change the name of the legend right in scale fill, because again, it overrides the previous argument. And this time you can change the color of the NA cells. So I changed them to gray. So now it looks like this, a little bit closer to what I was aiming for. And you can save this final tree. So to export the final tree, we first are going to save it to a new object. And then I just named it tree heat arbitrarily. And then we can use the ggsave function and save it to our output folder. You can also specify the DPI to improve the print resolution. If you have a very large tree, you can also, and you can only view it by using the zoom window. You can save that either automatically by right-clicking, but usually it might not have as nice of resolution. So you can specify the height and width, and it'll save as a very large tree, but it will maintain the DPI. And maybe this isn't great advice, but you can open it in Paint and scale it down, and it'll keep the DPI for publication purposes, which is nice. But that's pretty much the end of the road. Some of the basic steps that I use, and I encourage you guys to check out some of these resources, definitely the book. follow along some of the tutorials, and you can build and make some very cool trees closer to the publication one that we saw in the journal. Thank you everyone for attending. I hope you guys took up a couple tips away from this, and we can start the question period.